everyone who's joining the Masterclass today. My name is Lea Esser, and I am part of the research team here at Cube Biotech responsible for protein purification and activity assays. And I did my PhD just recent in the field of medical biology with a strong background in protein biochemistry. Today, I'm honored to host the Intelligent Infusion Masterclass from Cube Biotech, created for researchers to give them a platform and opportunity to share and promote their research with a worldwide audience. And we are so happy today to introduce our speaker, Andrew Lees, Dr. Andrew Lees. He will talk about conjugation chemistry and the construction of immunogens today. Andrew Lees is the founder and CEO of the company Fina Biosolutions, LSC Rockville, MD.
Fina Biosolutions is a conjugate vaccine R&D company, and we also manufacture carrier proteins and provide guidance in conjugation to our customers. I'm very pleased to be part of Cube Bio's Masterclass series so that I can share this guidance more broadly. Briefly, conjugate vaccines are vaccines in which a poorly immunogenic antigen is chemically linked to a carrier protein in order to enhance its immunogenicity. For example, conjugating a peptide, which is not immunogenic by itself, to a protein creates an immunogenic moiety. There are three elements to the conjugate: the antigen, the carrier protein, and the linker. We'll be looking at each of these three elements. But before you start your conjugation project, you should ask yourself what the purpose is, whether it's just to make an immunogen, which you're going to use once just to make antibodies, or use it as an ELISA antigen, or make an actual vaccine that you intend to eventually put into the clinic. In this talk, I'm going to be focusing on thio-ether conjugation. Specifically, the protein is derivatized with a thiol reactive group, such as a maleimide, and the peptide is synthesized with a thiol in the form of a cysteine. In happy circumstances, you just combine the two and your conjugate's done. All you need is a simple cleanup. In practice, however, it's not unusual to find the conjugation to be much more challenging. So let's look at all the components in more detail. First, we want to look at the solubility of the peptide. And this is the first thing we do when we receive a peptide, is we run it through a peptide solubility calculator. There's several on the web. The one we use is this one. You plug in the amino acid sequence and out pops a bunch of useful information. Notably, molecular weight and extinction coefficient, which are helpful for determining molar ratios, the charge, which will help guide you in optimizing the solubility, and a prediction of the hydrophobicity and water solubility. Now, these are only predictions, and you can use them as a guideline, but you should not treat it as absolutely true. You should plan ahead to have a linker on your peptide. If you don't, it can be very difficult to conjugate it without messing up your carefully determined sequence. For thio-ether conjugation, your peptide is going to have a cysteine. Ask for it in a reduced form. The cysteine can be on either the N or C terminal. If you don't know or don't have a preference or any reason to, I suggest putting it on the N terminal. I usually incorporate a spacer, CAGA, or on the C terminal, AGAC. And the ends of the peptide may be capped with, say, an acetate group in order to promote stability. You should note that anything you do, such as adding a linker to it, could affect your peptide structure, but it's very difficult randomly or offhand just to identify that. You should keep in mind that thiols will oxidize, forming a SS or disulfide bond. Oxidation is promoted by oxygen, of course, high pH, and metals. And to counteract that, you want to deoxygenate your buffers. Try to keep a lower pH, and I highly recommend the use of 2 to 5 millimolar EDTA to minimize metal oxidation. Every peptide is different. determining the suitable solubilization buffer may require some experimentation, and we use this solubilization rubric, starting with whether it's a shorter or longer peptide, acidic, basic, or neutral. We help determine at least a guess about how to get going. For example, an acidic protein, you may need to add a little bit of ammonium hydroxide as a base. For basic protein, adding acetic acid, you may need organic solvents like acetonitrile or DMSO in order to experiment to find a suitable buffer. I should note that some peptides and some people just standardly solubilize in a solvent like DMSO. DMSO in particular, you should note it's an oxidizing agent, and it will promote a disulfide bond formation. And if you store your peptide overnight in DMSO, you might find that all of your thiols are oxidized. So if you do make it up in DMSO, it's best to use it promptly. If you're going to store it in DMSO, you should freeze it. You might also try some alternative solvents like NMP or DMF. And make sure you don't use these solvents that have been sitting around for years and years, because there are some decomposition products that will also promote oxidation. Before you commit all of your valuable peptide to the conjugation project, I recommend that you first do a solubility test to evaluate whether your peptide is going to be soluble in the meeting. We'll cover that in the next few photos. So we store our peptides at -20 degrees. You want to make sure that you can find it again. We get a lot of peptides coming through. We label it and label what the purpose of the project, that is what protein it's going to be on and who it came from. After you allow it to come to room temperature, we recommend centrifuging it for a few seconds to get all the powder on the bottom of the tube. We prepare a clear glass test tube with 100 microliters of buffer. We take a 5-inch or some short glass Pasteur pipette and dip it into the powder to obtain a small amount on the tip. And then we put that into the buffer, twirl and swirl it, and then we take a careful look to see whether there are any particles floating around or if it's fully solubilized. These are some links to some other guidelines for solubility that may prove helpful. So you've purchased your cysteine peptide. I can guarantee you that even whatever they say for the purity of the peptide, if you weigh out 1 milligram, you will not actually end up with 1 milligram of peptide. There are other salts and things that are present that are not detected by the HPLC purity assay. So we use the DTNB assay to determine the actual thiol content, because what we care about are how many thiols, the group that will actually react with your protein, as the key determinant. So you've got your solubilized peptide. We use this colorimetric assay using the reagent DTNB or Elman's reagent. The reagent reacts with the free cysteine to give a yellow color, nice and visible. And you can use cysteine to create a standard curve. We'll use the extinction coefficient of 13,500 at 412 nanometers to get an estimate. And there's a link here for a protocol. But it's a very easy, very rapid assay, and necessary in order to make sure that you know what your actual thiol content is. Sometimes your peptide needs to be reduced, and TCP is the most common reducing reagent. You want to use a small molar excess over the peptide because you're going to have to remove it later. Now, TCP is, in the literature, it often says that it will not react with maleimide or another commonly used cross-linker, bromoacetate, but this is simply not true. It does react with both. After you do your reduction, you want to desalt using an appropriate molecular weight cutoff. And you should note resin, and you should note that most of the Zebra spin columns have a 7 kilodalton cutoff and are not suitable for desalting peptides. We now come to the selection of the carrier protein. Perhaps the most common carrier protein when you just want to make antibodies is keholipid hemocyanin, or KLH. It has the advantages of it's very large, it's a very foreign protein and highly immunogenic, has an abundance of lysines. The disadvantage is that it aggregates, it can be difficult to work with, it precipitates easily, and it's very hard to characterize the product. Another common protein is BSA, cheap, highly soluble, not easily denatured compared with many other carrier proteins, and but the disadvantage is it has relatively low immunogenicity. My opinion, BSA makes a better ELISA coating is better for making conjugates for ELISA coating reagents. Bacterial toxoids are often used for human vaccines. These include diphtheria toxin and tetanus toxoid that are treated with formaldehyde in order to toxoid them. Many of the primary means that are used for conjugation are blocked. These proteins tend to be heterogeneous, and it's can be difficult to characterize the conjugates for this reason. But they're also difficult to obtain, and if you can buy them, they're going to be expensive. Better than the chemically detoxified toxins are the genetically detoxified toxins, such as CRM197, which is a genetically detoxified diphtheria toxin. It's detoxified via a single mutation. As a recombinant protein, it's homogeneous. It has its full complement of lysines used for conjugation, and it's a proven carrier protein. It's one of the most widely used carrier proteins in conjugate vaccines. The protein has about 20 most accessible amino acids, or lysines rather. The detoxification causes the structure to be somewhat looser, and it has a tendency to aggregate easily. And under acidic conditions, the protein will sort of turn itself inside out and begin to aggregate, just like the diphtheria toxin. Fina Biosolutions produces CRM197 in the E. coli cytoplasm. We call it EcoCrem, both because it's produced in E. coli and also because it is one of the most economically well-priced CRM197 on the market. Notice our storage buffer. It's a low buffer strength heaps buffer at around pH 7.5. We have it in 10% sucrose to promote stability, and there's nothing in the buffer that will interfere with most conjugation methods. And having the low buffer strength means that the pH can easily be adjusted to whatever you need for your particular conjugation. Turning now to how we create maleimide proteins and how we label CRM197. We use NHS-activated carboxyls, or N-hydroxysuccinimide. These are reagents which will react with amino groups, allowing you to add a spacer to the lysine group. Among these NHS reagents are SMCC, shown on the left, and GMBS on the right. I would say SMCC is probably the most widely used maleimide reagent, but it's not one of my favorites because it is much more hydrophobic than GMBS. This can limit the amount that you can add to CRIM. Although the maleimide is somewhat more stable than the one on GMBS, the GMBS maleimide is in fact very stable. And another issue with SMCC is it has this large cyclohexane ring, which combined with the maleimide group is quite a large linker, and you do induce antibodies to that linker. There are, of course, many other linkers and combinations of NHS esters that are available on the market. You want to prepare your NHS ester in some product solvents, such as DMSO. We usually make it up in the range of 10 to 100 millimolar. You want to make it up concentrated enough that you're not going to exceed 5 to maybe 10% organic solvent in your final solution, but you also want to have it the volume that's easy enough to handle. You can freeze the reagents once they're solubilized in these solvents, but I prefer to make them up fresh. And I want you to note that even a small bit of moisture in the container will eventually hydrolyze all of it, and it's a catalytic hydrolysis. So the water molecules hydrolyzes one ester, and then it moves on to the next one. So eventually, the whole container will be hydrolyzed. We used to purchase in large amounts to save money, but found that before we used it up, it was bad, and we got unreliable results. So we now purchase in 25 to 100 mg units or some other convenient size according to the scale of our conjugation. To derivatize your protein with the NHS ester, you start with the protein, typically at 2 to 10 mg per mil. We keep the pH around 7.2, 7.4 so we don't hydrolyze the maleimide. You have your reagent prepared in your co-solvent. We're going to add between 10 to usually 40 moles of reagent per mole of creme. There's no point in overloading, trying to overreact the creme. You're more likely just simply to find it precipitating out, and you're really not going to get that much of your peptide on. I would add the NHS ester reagent slowly because it is in organic solvent and you're adding it to a protein. Gently vortex it while doing the addition, and I recommend perhaps checking the pH just to make sure that it hasn't dropped, because DMSO can cause the pH to drop. After 1 to 2 hours of reaction, you desalt it. You don't want this reaction to go on too long because the maleimide will, in fact, hydrolyze. So you desalt using either a spin device or a gel filtration column, and you end up with your maleimide derivatized protein. You're typically going to find yourself with 10 to 20 maleimides. This is a protocol from Thermo Fisher. The maleimide group is pH sensitive. In particular, as you get above pH 7.5, the half-life really becomes unworkably short. But as long as you're in the range of 7 to 7.5, you have plenty of time to work with it. And in fact, maleimide GMBS labeled creme, even after 3 hours, it's only decreased by about 5%. We like to determine how many maleimides are on our protein. We do this by having an excess of cysteine, allowing the maleimide to consume some of it, and then we go ahead and measure the unreacted cysteine using the DTNB assay, looking at the decrease or the consumed amount of cysteine, and using that to determine the maleimide protein ratio. The reaction of thiols with the maleimide group is quite fast. Even at pH 6.5, it can be a matter of just minutes, as shown on this graph. When you're working with larger molecules like peptides, it will react much faster than a small thiol like the mercaptoethanol, but usually the reaction is over within an hour or so. We're almost ready to start the actual conjugation. Before you commit your valuable peptide to the conjugation project, you should do a test conjugation. That way you can estimate the maximum amount of peptide you can add to your protein without there being precipitation. To do this, we will typically start with a known amount of protein, so say 100 microliters of your protein maleimide in the conjugation buffer, and add 5 moles of the peptide thiol per mole of protein. You want to do this slowly or dropwise and mixing while you're adding the peptide. Wait about a minute between each addition to see if the solution turns cloudy or opalescent. If it does, that means that's the maximum amount of peptide that you can add to your protein without it starting to come out of solution. So use slightly less than that for your larger scale conjugation. And now we start the actual conjugation. So you have your carrier protein labeled with the maleimide in solution. The protein is in the range of maybe 1 to 5 mg per mil and pH 6.5 to 7.5 hepes. It contains EDTA to minimize the thiol oxidation. You may want to do this on ice. You have your solubilized thiol peptide. You know the concentration of thiol, so that you know that when you add the thiol peptide to the protein, you can accurately determine the ratio of thiols per CRM 197. We would normally add between 10 and 50 thiols per crim. There's no point in going to a higher ratio because you're really not going to get any more on. And if you overlabel it, you may end up causing precipitation or aggregation. We let the reaction proceed for at least 1 hour, but we'll often let it run overnight just for convenience. Once the reaction is completed, you need to quench it. Quenching means that you block the active groups. We quench the thiol group. Now this is, I consider, optional because you're going to be removing the thiol peptide during the purification process. Using bromoacetamide, the reaction takes place at a little higher pH, so you raise the pH up towards in the range of 8 to 9. And then once that reaction is completed in about an hour, we add a maleimide quenching reagent, mercaptoethanol. This can be done at the higher pH, but we'll typically bring the pH down towards 7 again. However, we hardly ever actually fully quench both groups because the process of simply raising the pH up to 8 to 9 will cause the maleimide group to hydrolyze. So I feel like there's really no need to quench it. Next, we move on to the purification, done by any of the standard means, dialysis, a spin filter, gel filtration. You just want to make sure that you're using an appropriate cutoff dialysis tubing and spin device to make sure that the peptide is actually being separated away from your protein. Typically, anything over about 5 kilodaltons, most peptides are smaller than that. I recommend sterile filtering the conjugate, but before you do that, you should centrifuge it because there are sometimes little particles that will form. You can store it at 4 degrees. If you're going to freeze it, aliquot it to avoid repeat freeze thaw. Characterization, you can use OD280 or you can use BCA. Each of these will have some additional contribution from the peptide, so you want to take that into account. In some cases, you can make a correction because you can estimate how much peptide is on there using a method such as SDS-PAGE, where you look at the increase in the molecular weight and divide that by the peptide molecular weight to determine how many are there. Or you can use mass spec and looking at the increase in molecular weight over the molecular weight of the crim maleimide. We find that the SDS-PAGE gives a pretty good answer and is generally in in very good correlation with the mass spec data, and much easier to obtain. In general, should expect 5 to 10 peptides per crim, and there's usually no advantage to going higher than that. In fact, it may actually inhibit the antipeptide response if you overload it. And the larger the peptide, the lower the ratio you should expect, because there's steric hindrance that will reduce the amount that you can easily get on. Now, sometimes disaster happens and your conjugate, despite your best efforts, will begin to precipitate out of the solution. You can still run SDS page and show conjugation. You can mash up the conjugate to make a suspension of it. You can make a suspension with needles, and you can also beat it up with a bead beater and make little particles. Any of these, as long as you can get them into the injection syringe, through the injection syringe needle, you can still immunize mice for it. The peptide itself doesn't denature, and particles are often more immunogenic than proteins alone anyway. So you're off to do your immunization and your assays, and my part is done. If you'd like to learn more about it, you can find some more information in what's often called the Bible of Conjugation, Bioconjugate Techniques by Greg Hermanson. There's a lot of information online. Thermo Fisher in particular has a lot of information from their purchase of molecular probes many years ago. I do caution that, like this webinar, I can only offer general protocols. These may or may not work for your particular peptide, and it's always best to understand the chemistry so that you can troubleshoot your process. As I've mentioned, Fina Bio provides carrier proteins. These include not only the CRM197, but 8-MTT, a genetically detoxified tetanus toxin, a virus-like particle, as well as a tetanus fragment. We also have these proteins in a ready-to-conjugate format where they're already labeled with thiol-reactive groups like maleimide or bromoacetate. These really make the whole process of conjugation much simpler, because you can simply take your protein out of the freezer and you're ready to go. And with that, I conclude this presentation. I thank you for your attention, and again thank you, Biotech, for the opportunity to present to you. Thank you very much. So there were a few questions. Somebody asked about using DTT instead of TCP. You can use DTT, dithiothreotol, but in general, it's more convenient to use TCP. It's a more stable reagent. It doesn't smell, and you can use a lower molar ratio. And in all these cases, when you're doing your desalting step, you want to take care that you're properly separating the reagent, reducing reagent from your peptide. And so when we do these, we'll generally try to assay each fraction and make sure that we don't have any residual reducing reagent present using the DTNB assay. So let's see here. There was another question. Conjugate vaccine against TB, I presume that's tuberculosis. Well, if people have created polysaccharide conjugate vaccines, obviously you need you need your antigen. So if you have identified a protective peptide, then you could certainly go about creating a conjugate vaccine. The ones that I'm aware of, I think, use carbohydrates derived from TB, and that's usually a different type of chemistry, depending on the polysaccharide. And if it's something you're interested, I suggest you shoot me an email and we can discuss that. Next question. Let's see, only one cysteine. Yeah, if you have more than one cysteine on your peptide, then you are going to have multiple links to your protein, and it's quite likely that you will cross-link proteins, because one cysteine might react with one and the other will react with a different protein. So I suggest that if you do have a cysteine in the middle of your sequence, you might consider substituting serine for that, so that you have an OH instead of an SH. Generally, they are about the same size, but I would discuss this with your peptide company. In a few cases, people have sent us peptide sequences where the cysteine is in the middle, and we ask them not to put if it's okay to conjugate through that cysteine, we don't add a terminal cysteine onto it. So we just have one cysteine. Next question had to do with membrane proteins. Well, here the issue is solubility of your peptide. If it's a peptide that's meant to be found in a hydrophobic environment, chances are you'll have a hard time getting it into solution. And for these, if you're just trying to just induce antibodies, and the whole thing comes out of solution using one of those techniques that I suggested of trying to make a suspension of it and just injecting it, may well work. So, there are, not quite clear what you mean by conjugate vaccines. For polysaccharide vaccines, there are many. There are vaccines for pneumo and meningococcal, typhi. I don't think there are currently any human peptide conjugate vaccines that are on the market. I believe there are some veterinary vaccines that are made with a peptide. But there are certainly groups that are working on developing peptide-based conjugate vaccines, but nothing on the market yet. Any more? That's it. So if you do have further questions, you can find our email address on our website, and I usually respond pretty quickly. And we do offer not only the ready-to-conjugate peptides, but Fina Bio offers conjugate services as well. So if you're interested in that, just let me know.
So I think then thank you again very much, and also the audience for the questions. As you said already, the people are able to contact you through the email address on the homepage from Fina Biosolutions, and otherwise, due to the time, we should. We have another question coming in. So, yes. So one more.
Usually there's no need to use, gel filtration will purify it, so there's no particular need for an affinity tag. Most peptides are going to be in the range of 1,000 to 3,000 Daltons. The carrier proteins are typically, for CRIM, in the range of is 60,000. The tetanus is 150,000, so it's quite straightforward to desalt these on a gel filter with a simple gel filtration or spin device.
So then, thank you again, and then we should end this class for today, and the next masterclass will be on the 8th of October about the cryo-EM structures of cell-free synthesized PCR-G protein complexes, which is done by Dr. Frank Bernard from the Goethe University in Frankfurt. And he will say something, as the title induces already, about cryo-EM of cell-free synthesized GPCRs. And with this, thank you for joining the meeting. And again, thank you, Andrew, for giving this nice and interesting talk. And then see you at next masterclass.
Thank you.
Bye.
Bye-bye, everybody.